Welcome to this Data Analytics Practice Opportunity presentation session organized by the CHK Library. I'm Wendy Wong, the Research Data Librarian from the library. This presentation session is held both online and in person in our Digital Scholarship Lab. In this session, 3 teams of students participating in the Data Analytics Practice Opportunity 2021 and 22 will share their project outcomes. We are glad to have invited Professor King, Chairperson of Department of Computer Science and Engineering CHK, here on the screen, to give some comments to their presentations and work after their presentations. Audience here is also welcome to ask questions after their presentations. Online participants can type their questions or comments in the chat box of the Zoom room. Before the students' presentation, I'd like to introduce to you this event. The Data Analytics Practice Opportunity 2021 to 22 is held for the first time by the Research Support and Digital Initiatives team at the CHK Library. While 2 of the key areas in the CHK strategic planning 2021 to 25 are on student experience and research and innovation. The CHK Library also put education and student experience and research and innovation as 2 main areas in our action plan. Together with the growing importance of the research data management, which emphasizes the reuse of data, the Data Analytics Practice Opportunity was born. It aims to encourage the reuse of data, supports the exploration of real data from the library and university resources, promotes data mining and visualization, provides students with opportunities to develop data analytics practice and projects with real data, and encourage interdisciplinary exploration. We started the call for application in mid-December last year. Teams of 1 to 3 members were welcome. We received 26 teams of applications with 57 students. After the interviews in late January, 3 teams of students were selected. Among the 6 participating students, 4 of them are from Department of Computer Science and Engineering. 1 is from Faculty of Medicine and 1 from Department of Statistics. With the selected team, we started working on the projects from late January. The 3 teams have been working with Mr. Ryani, Head of Digital Initiatives, Ms. Kitty Seal, Digital Scholarship Librarian, and me. Throughout the semester, and all of us had 3 sharing sessions in the past half a year to share our findings, visualization skills, and provide support for each other. The 3 teams respectively work on 3 topics. Lek Wu, Juno Yang, and Steve Cheng work on the topic text analysis on collection, exegesis of recipe. Yifang Zapgai, Hangji Li, and Deyuan focused on the OCR and data analysis of the Hong Kong news. These 2 projects take data from the Chinese medicine test collection and the Hong Kong early tabloid newspaper collection from the CHK digital repository. John Yeung, we used the Millipede Genomes dataset deposited by Professor Jerome Hui in the CHK research data repository for genome assembly with the Bruijn graph. At the end of the project, each team has created a website, a poster, and a visualization tool. And they will also share their outcomes in this presentation session. With their great work and efforts, we supported each team with a stipend of $5,000. These are their websites. They will be further be linked to our digital scholarship projects site for public access. These are their posters. They will be printed and hanged in this digital scholarship lab in the coming year. That is over there. The visualization tool will also be put on our digital scholarship tools platform in the future. This is our working team. Other than Ryan, Kitty, and me, we also have our colleagues, Joe Cheng and Tyler Wen, to support this data analytics practice opportunity 2021 and 22. We are appreciative to have these 3 teams of students to make these events come true. We will continue our second cohort of data analytics practice opportunity in the first semester of the next academic year. The call for application will start in the late August, that is next month. The amount of stipend will also be increased to $8,000. If you are interested, please keep an eye on our library website and the CHK mass mail. This is the end of the introduction of the data analytics practice opportunity. Before I pass the mic to the students for their presentations, may I first invite Ms. Patty Zheng, Acting University Librarian, to present the students' certificate. First, may I invite team members on test analysis on collection exegesis of recipe, Steve Chen, please. Steve Chen is Year 2 students from Computer Science and Engineering. Juno Yao, please. Juno is Year 3 students from Faculty of Medicine. This team also have Nick Wu, Year 2 students from Computer Science. He is not available today. We are glad to see this interdisciplinary collaboration. Next, let's invite the team members on OCR and data analysis to Hong Kong News. Hangji Li, please. Hangji is Year 3 students from Computer Science and Engineering. Deyuan Khan, please. He is a Year 3 students from Risk Management Science program. Lastly, may I invite John Yeung, Year 4 students from Computer Science and Engineering. He is working on the Bruijn graph in genome assembly with Millipede genome datasets. Shall we all have all students and Ms. Zheng, as well as Professor King, to join together to take a big photo? Yes, yes, please. Thank you very much. Now, it is the most important part of today's event, the presentation. Let's first invite Juno Yao and Steve Wu to present their projects on the test analysis on collection exegesis of recipe.
Hello. So good afternoon, everyone. Thank you, Professor King, Ms. Chun, and everyone for joining our presentation today. I'm Steve, one of the team members for this project. Today, I am honored to introduce everyone of our project, test analysis on collection exegesis of recipes. So before we start, allow me to introduce you the index of today's presentations. So first, for introduction, we would like to demonstrate what we are trying to do in this project, or maybe an introduction of our target. Next, we would like to show our motivation for this project, why we would like to do this project for the public. And afterward, we would like to guide you guys step by step of our project. And lastly, in the reflection session, we would like to demonstrate the challenge we have encountered and maybe the future direction of our project. So our project will be aimed to do an ingredient usage analysis on collected exegesis of recipes, which is written by Wang in 1682. This series has included an impressive number, over 800 Chinese medicine recipes, divided in 21 chapters. Also, it has summarized the recipes invented by all the Chinese medicine practitioners in the past. Therefore, this series has extremely high practical value and contributed greatly to the development of modern Chinese medicines. For the motivations, we have found that the people around us usually don't have a very deep understanding of Chinese medicines. Therefore, we would like to promote the use of Chinese medicine via this project. Our proposed solution will be to represent the series in a modern technological style and also produce interesting statistics about the use of ingredients among different recipes. For the technology we have applied in this project, first, special thanks to Academia Sinica for providing the technologies and ingredient data sets. We'll be able to borrow the OCR technology from them and also a sample Chinese ingredient database. And also, we have chosen Python for our data extraction tool because of its diverse library support and also feasibility. Lastly, we have used Graphy for our test visualization tool, which is a popular graph visualization platform. For methodologies, we have divided the project into 3 important stages. They are OCR tests, data extraction and cleansing, and data analysis and visualizations. For OCR tests, we eventually choose to receive help from Academia Sinica because we have seen several problems in our targets, and therefore, after taking project capacity and the time we have into account, we choose to borrow the technologies from Academia Sinica instead of developing the OCR engine ourselves. For example, we can see here, the target may have inconsistent test patterns. Maybe they will only show 1 line of text here, or maybe 2 lines of text here. And also transparent background, which you can see some of the work was shown in the page behind, and also some marking, which will hinder the development of the OCR engine. And besides of the development of OCR technologies, we also prepare the data sets. We choose our data source from the digital copies of the test on CUHK library digital repository. And after receiving the first draft returned by the Academia Sinica, we have to perform a proofreading process and return the adjusted version. That is, if we found any mistakes in the first draft of OCR technology from the OCR test of Academia Sinica, we have to adjust it for enhancing the further scanning accuracy. Next, after we have finished the OCR test phase, we can move on to data extraction and cleansing. To set up the test scanning process, we have to first build up a keyword database for comparing the test and extract the key ingredients from the target. Thanks to Academia Sinica and also University Library, who has provided a sample database, this process has been greatly speed up. And after we have the keyword database, we can perform the following iteration. First, we'll perform a test scanning process. And then if we found the result is not very satisfying, we'll adjust the algorithm, or maybe we'll update our database, and then we'll go through this iteration again until we found the outcome satisfactory. And here is the code demonstration. First, in this code, we'll locate and scan the keyword in the test, and we have to make sure whether the algorithm will be able to locate the keywords and the number of the keywords are correct. And then after we have the result, we'll rearrange them into different CSV file for further data analysis and visualizations. And during this process, we have found some problems, like maybe some of the ingredients were not included in the database, or maybe some of them have different names, or maybe some of the recipes have different variations which are not included in the database. However, this problem cannot be simply fixed by adjusting the code. Therefore, we have to perform manual updates for our database. After the end of data extraction, we can move on to data visualization and analysis, which I would like to pass over the stage to Juno. Juno, please.
Thanks, Steve. So I'm in charge of the data analysis and also visualization. So in this project, as Steve has talked about, we are trying to visualize the relationship between different ingredients in different recipes. So our goal is to visualize such relationship in a giant graph network, which is an undirected graph network that uses the ingredients as the nodes and also establish an edge between them if they appear together in the same recipe. So first of all, we have to build a dictionary of all the ingredients, and rows of code will achieve this purpose. And we can also show code demonstration for how can we get the association dictionary. So the association dictionary is actually a storage of all the associations of the different ingredients in the recipes across the whole book. So these lines will allow us to pass through the whole book and then get the recipes and their corresponding ingredients. And then each of the recipes will be passed through these lines of codes to generate an ingredient association dictionary using the ingredients pairs as the keys and also their frequency of co-occurrence as the values. So let us move on to the graph visualization. So we have chosen to visualize our generated graph using GAVI. And there are actually a lot of algorithms that can achieve visualization. And we have chosen to use the Fredman-Reingold algorithm, which is a force field directed algorithm that kind of groups the nodes that are more connected to each other. And we have chosen to use a randomized initial spatial coordinates. And another point is that we have color-coded the nodes and also the edges with respect to their degrees, which means that the nodes and edges with a higher degree will have a warmer color like red, and the reverse will have a cooler color like blue. And for the nodes, this means that the node degree will revert to the frequency of appearance of that ingredient in the whole book, and the edges would be the co-occurrent frequencies of the nodes that it is associated. And finally, we have also generated an interactive graph display website using the Sigma.js exporter. So this would be an example graph that we generated and from one recipe. And as we can see, after performing the algorithm, the ingredients that occurs the most frequently will appear in the center of the graph, and the reverse will appear in the periphery. And we can see that here, gamchou, it is in the middle and it is red in color, indicating that it has occurred the most frequently in the graph. And as we can see here, the edge between gamchou and yansam is actually having warmer color than most other connections, which means that these two ingredients are very commonly used together. And another point is this is our interactive website. And as we can see, as the cursor points at the ingredients and you click on them, this will shrink the graph into the subgraph that the clicked ingredients links to. And here you can see that, for instance, the degree and the frequency. The degree actually refers to how many times that ingredient appears in the book, and the frequency will refer to the highest edge weight that it has. So for the data analysis, there are several directions. The first one is simply descriptive statistics of the data. And the second one is to take the Xing weight, which is the hotness or the coldness in Chinese medicine concept, into consideration. And the third one would be to cluster all the ingredients based on their connectivities across all the recipes. A point to note for point 2 is that the Xing weight consideration is only performed using limited amounts of ingredients because we do not have the entire Xing weight dictionary. So this would be the most simple descriptive analysis. And we have identified the 10 most commonly used ingredients, which are color-coded with respect to their Xing weight. For instance, gamchou here, the green color, is actually refers to a neutral property, and dongguai actually refers to a warm property, and blue color will refer to a cold property. And as we can see, 4 out of 10 most commonly used ingredients are neutral in nature, which may actually reflect the essence of Chinese medicine, that it is for harmonizing and neutralizing. And here is 10 most commonly used ingredient pairs. So as I mentioned just now, we also took Xingwei into consideration. And here are the example of two of the chosen recipes. The blue one is actually called Ching which in English means that it kind of removes the hotness from our body. And the red one is actually which means that it removes the coldness from our body. And let us focus on the red graph. And we can see that if the recipe is trying to remove the coldness from our body, actually it uses more hot ingredients. And for the other recipe, it uses a more evenly distributed ingredients. So this graph would actually show the final cluster of all the ingredients across all the recipes. So we performed a clustering using the Louvain algorithm, and this actually allows us to cluster the ingredients into different modularity classes. And then we use another visualization tool to kind of separate the groups for easier visualization. So this would actually, for instance, if we focus on here, this would represent a community of ingredients that are commonly used together. So now is the session of reflection. One of the most important problems that I think I have found during the data analysis project is that actually we have learned a lot of like visualization algorithm and also clustering algorithm. But one problem is why should we use the specific algorithms? For instance, I have chosen the Fruchterman-Reingold algorithm. It is because it generates graphs that have a better scaling, so it can provide a better visualization for us. But there are also other visualization methods. And for the clustering algorithm, I have chosen to use the Louvain algorithm. But there are also other algorithms like the K-means algorithm or spectral clustering algorithm, and all of them will actually generate quite different results. And this problem is not only important for us, but it is also important for researchers. Like, why do they choose the algorithm that they use? And I think this point is the greatest reflection that I have.
And also during the data extraction stage, as mentioned, we have to perform manual updates in our database to solve those problems we have found during the data extraction. However, if we were able to apply machine learning technologies during the data extraction phase, this problem can be avoided because by machine learning, it will have better feasibilities and it will be able to detect some problem among our expectations. But however, due to time limits, our current available technology will not be able to use the technology, but use the more simple solution instead. So maybe in the future, we develop the algorithm and make it more efficient. So this will be the end of our presentation. Thank you.
Thank you very much, Juno and Steve. Their project is very interesting and related to their work. And next, may we invite Deyuan and Hangji Li to present their work on OCR and data analysis, the Hong Kong news.
Good afternoon, everyone. My name is Hangji Li. My teammate is Deyuan, and I are going to give a presentation about our project, OCR and data analysis of the Hong Kong news. In this presentation, we will go through 7 parts, which are background, dataset description, post-optical character recognition, natural language processing, data visualization challenges, and our references. First, let's talk about the background of the Hong Kong news. The Hong Kong news started in 1941 after the surrender of the British colony. It records the undiluted voice and mindset of the Japanese administration of occupied Hong Kong. With the Hong Kong news, we can return to that period of history and have some ideas about people's thinking at that period. We believe that keywords in newspapers can reflect the trends at that time. So we decided to extract keywords from the Hong Kong news to get some information about the trends. The Hong Kong news data set contains 2,305 images distributed from July the 26th in 1942 to July the 30th in 1945. This distribution is uneven. which means some dates may have only 2 pages, while some other dates could have 6 pages or even 7 pages. The dataset provided by the library is the raw images of the Hong Kong news and the OCR results of the Hong Kong news. The raw data are in the forms of JPG files and PDF files, and the OCR results are in the forms of TXT files and HTML files. Now let's move on to our post-OCR process. First, what is OCR? OCR stands for Optical Character Recognition, which is a technology that recognizes text within a digital image. It is commonly used to recognize text in scanned documents and images. Post-OCR is some techniques to improve the quality of OCR results. At first, we wanted to directly utilize the OCR results provided by the library. However, we found that the accuracy of this result is not satisfying. Some Chinese letters even appeared in the original OCR result. As you can see in these red images, there is a Chinese character Kao in the original OCR result, and there is something that's not even English words here. Now let's talk about the methods we used in the post-OCR process. First, we used Tencent OCR engine, Ali OCR engine, and Google OCR engine to process the raw images separately. And we store the 3 sets of results in the forms of location and result pairs. The data structure we use here is simply a dictionary in Python. The keys of this dictionary are the location of the bounding box, and the values of this dictionary are the OCR results. Then we thought about reassemble each of the results into a long string according to the location information stored earlier. And we wanted to align the 3 long strings and compare them with each other to get the correct result. However, the 3 OCR engines have different ways to segmenting the images. Because we don't worry about the order of the sentences, we decided to do the text alignment and the correction in the unit of the sentence. In other words, we would correct the result sentence by sentence. Considering that the Tencent OCR result is in the unit of sentence, like this, and the other 2 results are in the unit of word, we use the bounding box of the Tencent OCR result as the basis and found the corresponding result of the other 2 results stored in this bounding box, like what you can see in the red images. In each bounding box, we align the 3 results of different OCR engines, and starting from the beginning of the sentence, we correct this sentence using voting policy, word by word. For each word, if any 2 of the OCR engines gave the same result, then this result was considered correct. If all 3 OCR engines give different results, then we simply use the result of Tencent engine as the correct one. For example, if the Google engine gave us the result of word, Ali gave us the result of word, Tencent gave us the result of word, using our voting policy, we would consider the word as our correct word. Now, please, my teammate Deyuan give more details about the later parts of our project.
Hello, Hangji. Thanks for the Hangji. I will continue our presentation for the rest of the parts. At first, let me do a brief introduction about my major topics, NLP. Natural language processing, also called NLP, is a subfield of the linguistics and the computer science, focused on how to program computers to process and analyze a large amounts of the natural language data. And the major topics in this area includes the tokenizations, paralleling, information extraction, similarity, and so on. And our project is more about the information extraction. And in summary, NLP relies on the computer to analyze the task semantics and the syntax, which can help drive the important information from the human language. And in our project, we only use some basic technique in NLP area to deal with the test data in the Hong Kong news to extract some keywords. And I divided our whole process into 4-step process. Pre-processing, back of words model and stop word detection and the TF-IDF index. Let me introduce the whole process at first. At first, with the raw test materials from the post-OCRs, But there is also some mistakes in these settings, except though they operate quality improves. And through the pre-processing, we have formed a normal sentence with correct grammars, and we derived the back of the word models to form a dictionary from those sentences. And after the stop word deletions, we compute the TF-IDF index to rank the words to form our final nano word list, the keyword list. And I have raised the funny sample to show you more about our process and help you better understand what we have done. As I shows, we have the room material test materials. I am a CA student sentence. It is obviously a wrong sentence with no means and with some special characters, extra commas, and a lot of tempos. So after the pre-processing, it can form a normal sentence like I'm a CHK student, and then we derived the dictionary of this sentence and divided each word and its frequency. But in the meanwhile, there is also some word like I am which is appears almost all the sentence, which is means it is meaningless to some degree. So we delete them and only have the CHK student, these two words. And then we want to calculate the TF-IDF index. But this index need to more than one sentence to compute. So that with another sentence which is randomly I'm a student, we will compute the CHK as 0 and the student as minus 0.2. So that we can finally get the keyword as CHK, which is the top one keyword in that sentence. And I have to mention that during the pre-processing and the stop word deletions, we only use the normal method and we adjust the algorithm again and again, which is almost tedious work. So that you can view the details in our website, and I want to skip these two parts at this presentation, and I will focus more on the bag of word models and the TF-IDF index. What is the bag of word models? Obviously, it is to build a dictionary and count the number of the occurrence of words for a given text in the lexicographic orders. And we use the counter vector package in Python to transfer the pages text into corresponding word bags, also called word dictionary. In each box, it stores the word as features vector and the word's frequency as a values vector to facilitate the next step. You can see from the right pages, and it will show you more information clearly. And about the TF-IDF index, I want to introduce the two basic definitions at the first, which is the term frequency. The term frequency is the term frequency of term is the ratio of the number of times the terms appears in a document compared to the total number of the words in the document. And the computer process is shown at the right side, and there is also the IDF is called the inverse document frequency it reflects the proportion of the document in the corpus that contains the terms. Word which is unique to small word of document received higher weights than the word appearing across all the documents. And its convener function is also shown at the right side. And then we want to combine these two indexes and form our final criteria, TF-IDF index. It is simply that TF multiplied by the IDF. And our target is to extract the words which are more frequently in documents, but also have high freshness, also called the low popularity. This is the key criteria to find our keyword. And with the keyword, we can operate the finally these visualizations. And our visualization target is that based on the extracted keywords, we want to process the trade of the changes of keyword with the time going by. Meanwhile, we are also willing to supply a convenient method to search the material about the keyword, which can facilitate scholars who want to search, who have the research request towards the Hong Kong news. And for the method, we construct an interactive bar chart with a draggable timeline to identify our specifications. and clicks the keyword bar. The link of the link to pages represented by the keyword will be shown below. And I also raised the examples to help you better understand. For example, if a scholar wants to search the attitude of the Japanese administration about India's war from the 1942 to 1944, he or she can drag the timeline to facilitate the trade of frequency of the keyword India's. If the scholar is interested in some in news related to India's in some specific period, he or she can click the keyword and can simply find all pages represented by that keyword. And let me demonstrate the details. What? Yeah, as you can see the top 50 keyword in the all-hill period. And we randomly choose the word duck. It shows like the 12 rings in all of the 50 words. And we can drag the timeline and you can see what happened. Oh yeah, the duck is come to the medians, and if we continue to drag the timeline, and the duck is disappeared. So what happens? I think it may be the interest in some scholars to research about, and if they want to research about that, he can search the duck. He just click it, and he can find all pages which has a keyword about the duck and search what happened because it disappeared in later period. I will show you. Just click it and you can find the real pages at that page. And that's all for our visualization, and let's continue our presentation. Actually, our presentation also have some limitations because about some technique issues. About the accuracy, because we don't have the ground truth about the OCR result, we can calculate the accuracy of our post-OCR results. And about the alignment, actually we want to form a correct test for contents in the Hong Kong news. But we found though the accuracy improves much, it is still some sentence which is hard to form a correct sentence with clear meanings. So with our target just to extract the keyword, we only choose the sentences which have better quality to continue our analysis. Maybe with some machine learning method, we can finish that the alignment the process to form some materials much useful. And this is all for our presentation. Thanks for your listening.
Thank you very much, Hangji and Duran. Their tool will be very useful for the scholars to do future research in the future, I'm sure. And next and finally, may I invite John Yeung to present his work on the Bruijn graph in genome assembly with Millipede genome dataset. John, please.
So I'm John, and I'm working on a project called the Bruijn graph in genome assembly with the Millipede genome dataset. So why do we need this kind of work? Why do we need to do genome assembly? So to the majority of the public, they may not know that. So suppose there are some products in the market that allows you to send your blood and let the association to with your DNA and see if you are allergic to something, if you have some diseases. But this process is not done by magic. So does not exist a machine that after you put your blood in it and you can read the entire 30 billion strings in your body just at once. Instead, we obtain what's so-called short bits, which are subsequences in the DNA of any kind of body, maybe RNA. but I'll use DNA example here. And to generate the complete sequence from the obtained short reads, so short reads are short sequences, we need to have an algorithm that help us to do it fast and accurate. And here comes the Deyuan graph algorithm. Usually, the short reads that we can obtain is about 100 to 200 characters long. And what we want to do is have these kind of different short reads and overlap them together to construct an entire complete original DNA. And this product is what we call context. So it's just some keywords. Context is the final product we want to achieve. And then this kind of context will be given to researchers in the field of biology for further research. So one quick example, we have these two short reads, and you can observe that they have the same overlapping area CGGAC and CGGAC. And we want to overlap them together and construct this complete sequence. Of course, in real life, the numbers and the length are much higher. Just a quick example. And we are using the Millipede genome dataset, which is used by Professor Cheng. We also thank him for allowing us to use this dataset. The dataset is used in this paper, which can be retrieved in the CHK research data repository. We attempt to implement this algorithm and use this dataset to demonstrate how this sequence assembly process. In this demonstration, we use the first 100 samples because if the numbers go high, the constructed graph and the representation will be too cumbersome. And for demonstration purposes, we only use the first 100 samples. And we use the samples from this file. So this is a breed of Millipede. So just like dogs, we have golden retrievers and something like that. This is the same for Millipede. And the data file format is like this. Oh, sorry. This is called the first state data format, just example. And we have an arrow, we have the name, and perhaps some descriptive text at the optional descriptive text at the end. And follow-up will be the sequences. And just keep going on. So this, let me hide that. So this is a screenshot from the actual data file. You can see it has an arrow, has a name, and maybe some text. It doesn't have it here. And follow-up is the entire short read, or what I hope it was. But it was, so here, this is the actual data. It actually goes way below, but there's not enough room to show it. So so also the reason I did not show it here. Okay. Hello. Okay. So how does the Deyuan graph work? Before that, I would like to answer a question: why not other algorithms? So one very simple algorithm is to do brute force tracking. So what we want to do for two sequences, we want to do a styling window, and if we see an overlap area, we merge them. But because the enormous amount of data, we cannot do it directly because it's too slow. And it may take us ages to do it. The other method is what's called the overlap graph. I'm not going into details here, but simply speaking, it constructs a graph in a mathematical sense. So we have some nodes and edges. But using this graph could fail in some edge cases. So in modern days, the sequencing machines only use the Bruijn graph algorithm, which is fast and more accurate in some cases. So a better alternatives than these two. So how it works is that we can construct a graph with hyperparameter, which is a user input value called K. And setting a K can allow us to use different K-mers. So K-mers is a term used to describe the length of the subsequences that we use to construct the graph. And then we can apply a Eulerian path algorithm on it to trace back the graph and obtain the final sequence. So simple example, we have ATCG, which is the 4 characters representing the 4 different acids in our DNA. So suppose we have this short width, and we are using K equals to 3. So we extract the 3 mers. So first we have ATC and TCG. And then because TCG is followed by ATC, so we add a directed arrow to it. So usually we will have a circle around it to create a node. And this arrow we will create a direct edge. So this is a graph, the Bruijn graph formed by this short with ATCG with K equals to 3. Another example, we are using K equals to 4 here. So very simple. There are 2 arrows here because there are 2 TCGG and CGGA. So TCGG, CGGA. Okay. CGGA, UGA. Okay. Maybe not. I just want it. Okay. So you get the idea. And this is another example. So this is correct. Generated using a computer, so shouldn't be any human error. So you can, after we have this, we can use trace it back. So TCTT, that TCTCGCTACTCCCG, and we can. So this is actually the constructing sequence here, and similar for the below one. And we can. So after tracing back, we can obtain the 2 complete sequences. And a more complicated example. So this is also a deep Bruijn graph with K equals to 12. So what is the results when we apply it to the Millipede genome dataset? It generates a corresponding Bruijn graph. We can use it to trace back and construct a fitting sequence. So we can construct a context from it. There's 2 demo videos with different value of K. But it's only constructed using 2 shorter sequences in the dataset because if we use more, the graph will be too cumbersome. So this is generated using K equals to 37. So afterwards, it becomes 1 graph. But you cannot see the relationship between the 2 sequences because they are all coupled together. But if we increase the value of K, with this one is K equals to 49, at the end, it will be separated into 2 sequences because there is not same 79 mers in this data set, else they will be linked together and we cannot see its differences. And if we map the first 100 shortlist results from the dataset to the obtained shortlist. So above is what we get from the dataset, and below is what we get after the algorithm. If we focus on the first three shortlist and content mapping, we can observe that they seems to all have a one-to-one mapping. Why is that? That's because the dataset that we obtained is actually not the raw data. Instead, they are already assembled genome data. So they are already the context. So that's the reason why we do not have the similar graph I showed at the beginning, like this. So which is also true for other the subsequent shortlists. But still, we can make good use of this, and we have a website describing the our project in detail, and we also have an interactive tool that allows users to visualize our sample or custom data sets from the user with treatable hyperparameter K. So given a sample, we can generate a corresponding Deyuan graph. And you can also play with the hyperparameter value of K and see how it affects the visualization. And we also have the mapping. And for a more complicated example, 4 is too low. Okay. Let's wait for a bit. And for longer sequences, you can also generate via this visualization tool. Let me show you the mapping. So something like this. And you can actually see what is going on under the hood. And let's switch back to the PowerPoint. So with this, I believe you serve as a great tool for biology students, engineering students, and most importantly, bioinformatics students who is studying how we can use computer science on biology data. And so this will be the end of my presentation. Thank you all for listening.
Thank you very much, John, for his interesting presentation. I think that would be a very useful tool for educational purpose, especially for those in the field, in the biological, bioscience field, as well as someone not really in depth to genome, that I can understand more about genome now. Now may all the students come up to the stage, and we would like to invite Professor King to give some comments to the students' work. If online audience would like to ask questions or give comments, you may also start writing your messages in the chat box. Now Professor King is on our screen. So you can, Professor King can give some conversation or some comments to our students.
Okay. Well, first I'd like to just thank you and other colleagues from the library for inviting me to this wonderful event. I think the work from the students are quite excellent. I'm actually impressed by what they have done. Obviously, there could be more things to be in each the project, but given the time and the knowledge that they have done a very, very good job. I would just say that this is really an initial stage, so a lot of things are still can be explored further. I would like to see from each of the project some of the good observation and predictions that they actually can make. And maybe they could even, for example, the last one on the graph, they may be able to predict maybe some diseases or other things if it is connected with other database together. And other projects as well. I think there are many future work that can be done. So I look forward to even more from other students who will be interested in data analytics and do this type of research. I think that it really encourage people to think deeply about computational thinking, and apply what they know to do well. Because you see that there are three different things: one in medical, one in news media, and one in two in medical, basically recipe and the graph. So these are really excellent work. So I really appreciate it. I'll send you some of my text written notes a little bit later. But that's what I had to say. And thank you again for this wonderfully organized event. Thank you, Wendy.
Thank you very much, Professor King, for your comments. And we do appreciate your comments, especially that you tell us that the projects can be further developed. And we do hope in the next year, some students can come up to continuative projects and do further research on that or development on this types of data. So for now, would the audience in this room, in the DS lab, have any questions or comments to the students? Or online audience, do you have any comments? Maybe someone is writing. While someone is writing, I would like to ask the students here on the stage, can you, from each group of you, can you share your how you think about this project? You have been working on this project for around 6 months, starting from end of January. So maybe can you share with us how you think about this project? What you learn from this project? Maybe would it be useful to your future or future study or future career? And I can pass the mic to you all. And
Testing. So one of the things that I think is after joining this project, we have tried to analyze a lot, kind of a big data set. And I have faced a problem is that even if I visualize or performed clustering on the data, actually I'm still not sure about the underlying meaning of the clustered data. And I think that this is also a common theme in a lot of biological fields, like for instance, in the metabolic pathways, maybe they can cluster a lot of proteins that kind of work together, but what is the actual importance of that? And maybe in neuroscience, it is the same. They cluster different neural signals and kind of assigns different groups of neurons to different tasks. But again, what is the significance of that? And I think that this is one thing that we should bear in mind when dealing with big data sets, is that we kind of, even if we can identify some of the correlation, we should always try to think about the underlying meaning after the analysis and also during the analysis.
thank you, Chun.
So I also working on this project, I really feel I was given the opportunity to try out different algorithms and maybe some visualization tools that I've never heard of or used before. So suppose in this project, I used a package in Python called polydash that allowed me to construct and visualize the Bruijn graph. So I really think this will be a great experience for me, and for the data set, I really appreciate how different people are collaborating together to give information about how to use and cite different kind of data sets, which really benefits me for my further studies or even research in the future.
We really appreciate for our group. We really appreciate the library for providing us this opportunity to practice more of what we learned from the courses. And this is an opportunity for us to do complete works from the design of the project to the implementation. And we learned a lot from the implementation, such as something about OCR and the NLP process. Well, that's all.
Thank you very much for your sharing. I hope your sharing today will be useful to other future comers to our data analytics practice opportunity. And any comments or questions from the audience? If no, may I give a little bit feedback or maybe some food for thought from what I've just heard from their presentations. Just now when John, I think John and also Juno's group also mentioned about why you choose such an algorithm for doing such a project, right? So I think while we are doing data analytics, data analysis, there are a number of algorithm that we can choose. What I think from their presentation is that they also raise a very important topic nowadays in data analysis, that is which algorithm do we use, and why do we choose such algorithm, and the bias between different algorithms we have. So I hope this can also give a message to computer scientists or data analysts in the future. Do you all have any comments or feedback to this area of topic?
Thank you. And I actually don't have any further comments, but I really encourage students or any people from the public to really take the chance to learn more about data science and analytics, because it plays a more and more important role in our world, and maybe in the future this will be included in math courses too. So it will be a great chance if you can take this opportunity and enrich yourself with this kind of knowledges. So this program will be a great opportunity for anyone with the willing to do so.
Yes, thank you, John. If there are no more feedback, may we end this session here? Thank you very much for coming to this session in person and online. And thank you very much for Professor King to joining us today, as well as and very important to Professor Zheng, who would like to come today, but was in a meeting and in a hurry and still not yet to arrive yet. And also to Academia Sinica for their support throughout this project. Thank you very much. Hope to see you in the next Data Analytics Practice Opportunity presentation session. Thank you.